Good afternoon. My name is Scott Friedland. In the next 3 minutes, I'm going to blow your mind about what goes on inside of a nucleus. Give you the world's briefest lesson in chromatin biology and tell you how this relates to one of the deadliest diseases that we face today. This is pancreatic cancer in the tale of the broken librarian. This story starts with you and your 30 trillion cells. Let's take one of them, crack it open, pull out the nucleus. Crack that open, pull out the DNA. Line it up end to end. It's 6 feet and has 20,000 genes. How did that fit into something that's 1/millionth its size? The answer? It's wrapped up. First, take DNA, wrap it around a protein called a nucleosome, and then wrap that around each other again and again. Throw in a couple more proteins, and voila, your genome as chromatin inside of a nucleus. But we need to be able to read our DNA. How are you going to do that if it's all glommed up in these proteins? To understand this, we're going to imagine the nucleus as a library with DNA. DNA is information stored as genes, which are basically how-to books for making cells. Pancreas cells need to read pancreas books, brain cells need to read brain books. But how do they know which of the 20,000 books they're supposed to read? The answer? All cells need a librarian. The librarian for the cell is called the switch-diff complex, whose protein member ARID1A is what I study. The switch-diff complex can take nucleosomes and move them along the genome in order to make more or less space, making genes more or less able to be read. As it turns out, the switch-diff complex is broken in most types of cancer, and ARID1A is mutated in about a fifth, including pancreatic cancer, the fourth leading cause of cancer-related death. To be diagnosed with pancreatic cancer means to have a less than 10% chance of living for the next 5 years. Terrifying. When the switch-diff complex is broken in pancreatic cancer, it doesn't just stop working. The librarian doesn't quit. Instead, it gives preferential access to genes that are important to cancer. In my work, what I found is that ARID1A is necessary for normal pancreatic development, that pancreatic cells without ARID1A proliferate unendingly, they divide unendingly, a hallmark of cancer. But most interestingly, we found that while these ARID1A mutations are beneficial to cancer, they may also be an Achilles heel, as these tumors are more susceptible to specific chemotherapies. By studying the switch-diff complex, the cellular librarian, our lab has given an opportunity to take the insights from molecular biology and apply them to clinical practice, making what is one of the deadliest diseases a little bit more manageable. The end.